Hi everyone, my name is DiemQuynh Nguyen. I'm a second-year PhD student in the Earth and Environmental Sciences lab, or department, I'm sorry. And today I'm excited to share with you the world of ants and their gut microbiome. And so to start, many animals have associated microbiomes that either supplement nutrients, neutralize toxins, and or shape host physiology and fitness. In this illustration here, don't worry too much about the tiny text. It's just here to showcase that we see this occurring in many systems, like with bumblebees, where they essentially metabolically break down plant carbohydrates as well as detoxify dietary compounds. In Drosophila, we see how their gut microbes affect the host's lifespan and fecundity. And in cephalotes turtle ants, because they have a herbivorous diet, they require on their resident gut microbes to partake in nitrogen recycling, as well as convert vertebrate excreta into usable amino acids. And so an interesting system to dive deeper into insect associations are the honeypot ants, which are an extremely diverse group. There are 8 independently evolved genera spanning across 3 subfamilies within the Formicidae ant family. But the one that we're going to be mainly focusing on today are the Myrmecocystus honeypot ants, which inhabit arid regions within North America. And so with these ants, within a colony, you have the queen, you have workers that go out and forage for food, and then you have these really specialized workers that you can see in the image here called repletes. Essentially what they do is they store these nutrients that they collect in this organ called the crop that resides inside their gaster. And then when resources are scarce, what they do is that they take the contents within their crop, regurgitate it to their fellow colony members, and this process is called trophallaxis. And so you can think of them as like living refrigerators out in the deserts, but for ants. And so this leads us to questions like, how do repletes preserve nutrient-rich fluids for extended periods of time without spoilage? Do Myrmecocystus honeypot ants have a resident gut microbiome? And if so, what is the relationship between the honeypot ant and their gut microbiome? Before we can really answer any of these questions, we have to start by asking, what is the taxonomic composition of the gut microbiome within Myrmecocystus honeypot ants? And so to do that, we went ahead and collected several species within Arizona and California. And then we went ahead and dissected the crop and midgut from these ants. And then we did DNA extractions on our gut samples, did PCR and gel electrophoresis to verify its amplification, sent our samples to UConn for library prep and 16S sequencing. And then when we got our sequences back, we processed our sequences and analyzed them. And so this is what we know so far. When looking at Myrmecocystus mexicanus within this species, when we look at the relative abundance across the replete crop versus replete midgut, comparing that to worker crop versus the worker midgut, we see that in replete crop, there is this dominance in this genus here in gold, and that's Fructilactobacillus. When you look at replete midgut, there's also Fructilactobacillus in there, but you can see other bacteria that are in the midgut, like Vibrio or Pseudomonas. And so we wanted to look at the other species. When we look at Myrmecocystus mendax, for that one, it seems like it tells a slightly different story. So in the replete crop, you don't see that abundance and dominance of Fructilactobacillus like you saw earlier in gold. Rather, you see this dominance in this red genus here. This is Zymobacter. And then when we look at other species like Myrmecocystus wooleri, that one, it doesn't have that same pattern where the replete crop is completely dominated by a particular genus. It seems to be a lot more variable. And so with this in mind, we went ahead and compared the alpha diversity between the crop and the midgut across the different species. So here in each plot, it represents each species. And then within those plots, we have our box plots comparing the 2 organs. So the crop is in orange, and then the midgut is in blue. And then what we see is that with Mexicanus and mendax, with these 2, it showed that there is a significantly higher diversity in the midgut compared to the crop. But when we look at the other species here, it didn't really show a significant organ level difference in diversity. We then wanted to compare or see how microbiome composition differs across species as well as organs. And so here we have an NMDS plot using weighted unifrac. Color-coded here are the species, and then the organs are denoted by different shapes. So with the crop, it's in circles. If it's a midgut, it's in triangles. And so points that are closer together means they're a lot more similar to one another. Points that are farther away means they're a lot less similar to one another. And what we see is that most of the samples, they cluster by species, with the exception of mexicanus and Woolleri. So mexicanus is in the light blue, Woolleri is in the dark red, where there is some sort of separation and clustering of the crop in the midgut. And that was statistically proven with Permanova and Anderson. Another thing to note here is that you can see that there's two distinct clustering of like these warmer tone colors and then these cooler tone colors. And so the warmer tone colors are the ones that are the samples that we collected from California, and the cooler tone colors are our samples that we collected from Arizona. So we do think that geographic context could play some role in influencing microbiome structure, and it seemed to be statistically proven with our Permanova and Anderson. And then, as I mentioned earlier, there are some species where the crop and midgut, they do seem to differ from one another in terms of microbiome structure. So I was curious to see which bacteria taxes were really driving the difference between these two organs. So to do that, I went ahead and used ANCOM BC to really see which genus was enriching which organ. And here we see that Leuconostoc, Zymobacter, Acetobacter, as well as Fructilactobacillus, those tend to be significantly higher in the crop. And then when you look at the midgut in which bacteria was significantly higher in that organ, we see that Vibrio occupies it, Testinobacter, and a few others. And then I wanted to see how prevalent these taxes were across our species and samples. And so when I looked at Myrmecocystus mexicanus, there was a prevalence of Fructilactobacillus across 98.9% of our samples. And then when we look at Mendax, 91.9% of our samples had Fructilactobacillus along with other bacteria. It's also interesting to note that the ones that we collected in California, like Myrmecocystus arenarius or Flaviceps, those ones seem to have holophilic archaea that occupy them. So it'd be interesting to dive deeper into that later on in the future. And so, yeah, so some key takeaways are Myrmecocystus mexicanus and Mendax, they show strong organ level compartmentalization. Myrmecocystus microbiomes seem to be species specific, and that core taxes, they seem to differ by species as well as possibly geography. And some things that we're planning to work on and do afterwards is we saw how Myrmecocystus mexicanus had that dominance of Fructilactobacillus. So we wonder if Fructilactobacillus could be involved in some sort of nutrient preservation, because this is a type of lactic acid that we commonly see in fermented foods to help prevent spoilage, like the sourdough that we eat or the kimchi that we consume. So moving forward, I'm planning to do some microbial tracking with green fluorescent protein to monitor where this bacteria is in the digestive system of these ants. And with that, I want to thank TSS for allowing me this opportunity to present, as well as my advisor, committee collaborators, and lab mates and peers. Thank you.
Could some of these microbes be producing a secondary metabolite that's like potential, maybe that's what they have in common? So although you go to different geographic locations and the microbes differ, could they have like possibly a physiological response or produce the same secondary metabolite which inhibits the growth of other microorganisms from spoiling the products that are in that, I want to say midgut or whatever the storage capsule is?
Yeah, it's definitely possible. I'm not too sure on really how to dive deeper into that, but it'd be interesting to discuss it with you.
I was curious whether or not you could look at treating these ants with an antibiotic that would get rid of the microbiome, and would that then, I guess, alter, I guess, the ability to provision these nutrients. You know, so in other words, if you get rid of the microbiome and you hypothesize that they're important for preserving it,
Yeah.
getting rid of those microbes would have a detrimental effect, right? So can you test that in more of a laboratory setting?
Yeah, I definitely think we should test that first to see if they exist in like a commensal relationship with these microbes, or they do rely on these microbes for more than even nutrient preservation. So, yeah.
Thank you.